Hay algunas cosas de esos planetas que sí nos afectan cotidianamente y uno de los equipos que va a presentar ahora de TecnoX, el equipo de Brasil, nos va a hablar de cómo nos puede afectar Mercurio en nuestra vida.
Malísimo. Muy mal el chiste.
Hey guys, good morning. My name is Anderson, these are Maria and Lais, and we came from Federal University of Amazonas to show you our project, Vídeo, an innovative method to mercury bioremediation in E. coli. But why this method deserves so much attention? Mercury is almost everywhere. In nature, it's found in several forms and can pass through a process called biomagnification. When it's absorbed by algae, then feeds small fishes, that feeds larger fishes, increasing the concentration of mercury in its tissues until the man consumes these fishes. And in our region, in Amazon, we eat more fish than all other Brazilian regions. And it's claimed that there are about 3 tons of mercury in our rivers. More mercury than places that suffered an accident like Minamata Bay or Iraq. And how did we achieve these high levels? In Amazon, there are huge gold exploration and mining. And how do you separate the gold from the clay? With mercury. That it's terrible because this increased the concentration of mercury in our rivers. And believe there is no regulation to this manipulation. And there is information from this year that in a native population of the Yanomami, 92 is contaminated with mercury, which can cause neurological disease and plenty of other health problems.
And how can we develop alternative solutions to this problem? Why not use synthetic biology as a tool? Yes, in vivo, the reason we are here. Bacteria are capable to grow in different environments, including the toxic ones. We found a group of genes involved in mercury resistance in several bacteria called MER-operon. And how this system works. In the presence of AG, it is linked to bacteria inert by transporters protein MER-P and MER-T. There, it leads to MER-A, mercury reductase, reducing ion AG2 to AG0, a less toxic and volatile form, getting out by passive transport through cell membrane. Other important fact about the MER-operon is its regulation. It is just an awesome system. Take a look. In the absence of AG, MER-R repressor protein binds to the operon promoters regions, blocking the RNA polymerase recognition and this way the transcription. But in the presence of AG, MER-R repressor doesn't resist to AG's seduction power, so leaves the promoter. This way the RNA polymerase can recognize the promoter and the MER genes can be expressed. So only in the presence of AG, the genes will be expressed, okay? We utilized this beautiful mechanism as inspiration to develop a new genetic construct to improve the mercury bioremediation in genetically modified bacteria. So, our first construction was to design a new promoter to increase the MER-operon expression. So, we designed a new handsome promoter that looks to RNA polymerase and say,
"Hey sweetie, let's make some pretty messages RNA, Tom, you and me."
Of course! And to do that, we put together the sequence of tap promoter, which is a well-known and widely used promoter, to the sequence of mer operator. So, resulting in a new promoter with the both sequence. And how can we measure the promoter's efficiency? Everything looks cool with fluorescent proteins. So, we linked the new regulated promoter to the red fluorescent protein and measured by the emitted fluorescent level in the presence of AG. And there are our pretty results. Here, E. coli expressing RFB under control of our new promoter without the repressor protein, so in a constitutive form, with huge fluorescence, so it's a strong promoter. And here, our final construction confirmation analyzed by enzymatic digestion test. And here, the expression difference with the repressor and without, and we achieve a clear difference. It's like repressing and in a constitutive form. So, with clear difference. And here the RFB with our repressor and without the repressor, and after 4 hours measured by spectrophotometer, and after 4 hours the bacteria without the repressor had 230% increase the fluorescence. So, showing its potential of our new construction as a regulated promoter by our repressor. Now, Lais will show the second part of our project. Thanks.
A second part of our project was the construction of the mer operon, and we did two options. In the first, there is the mer gene that produces a regulatory protein to mercury. There is genes that produce a mercury transporter proteins, MERT and MERP, and a gene to mercury reductase, MERA. Here, there is a bidirectional promoter that do the transcription in a constitutive form in the left side, the MERA direction, and in a regulated form to the right direction. The difference between our second and first construction is the addition of the MERB gene in the second construction that produces a protein that can break down methylmercury. So, when there is mercury in the medium, there are the transcriptions of the genes in the right direction of the bidirectional promoter. So, to beginning of test, we did an experiment using our first construction in media with mercury, and here we can see that in 5 ppm, after 1 hour and a half with the addition of mercury, the control bacteria didn't grow in time. It starts dying, while all bacteria with our construction is growing in the time, showing the resistance of our construction in bacteria in mercury. So our bacteria could grow almost 5 times compared with the control. After we did a second experiment, an experiment of inhibition zone. So we used discs with mercury in the plates, and here a negative control bacteria, a bacteria with our first construction and with our second construction with the addition of MERB gene. And we can see that with 2% of mercury, our second construction presented a lowest inhibition zone, so the bacteria could grow nearest to the discs, and the inhibition zone increased in our first construction and in our negative control. So inhibition zone reduces 30% of a first to second construction bacteria. Here, in 200 ppm, the inhibition zone reduces 39% of a first construction compared with the control here, and 6% in your second construction. And in a concentration of 20 ppm, there is an inhibition zone in your second construction, and a little difference between your first and control. So second construction showing the potential to be used in bioremediation systems with mercury. Now Anderson will talk about our outreach project.
So this exciting project could be only at lab. So to return to science our results, we developed innovative methods to teach SimBio in school with some practice. Before we get started with the lectures with high school students, we applied a survey, a questionnaire, because we wanted to know if they ever heard about synthetic biology-related things, such as genetic engineering and transgenic. And as we can see in this panorama, this overview, in graph Q1, from the 111 attended students, only 8% ever heard about the synthetic biology-related things at science classes at school. And in graph Q2, we can see that 39 did not believe that it was possible to genetically modify a living being before our lecture. And only 18 knew how to identify a living organism genetically modified. And to solve this problem with the high school, the science classes in high school of our city, we used neon bracelets to symbolize the plasmids used in our construction with the GFP gene and the mercury binding protein genes. So these models of plasmid were inserted in bacteria models that accidentally looked like minions, and they symbolized our genetic constructions from 2014, and they had magnets in the surface to symbolize the bioaccumulator, and we used screws that would get attracted by the magnets as a bioaccumulator would do with mercury. And the second model was to represent the biosensor. They had LED lights inside with the poles to the upper side, and a polystyrene ball with a battery inside representing mercury. So when our bacterial model poles leads to the poles of our mercury, the lights were on as the biosensor would do in the presence of mercury. And here our incredible team and our advisors, Carlos, Gustavo, and Espartaco. We are pretty thankful for the invite of the organizers, thankful for your attention, and I'm especially thankful for our friends, especially Giovanna and Maria Leticia, my other hands. And here are our sponsors. Thank you very much.